Biology, My Passion. I am Soumya Harikrishna. We are now learning the chapter Molecular Basis of Inheritance. We discussed up to central dogma. Today we are going to learn about DNA replication, which is actually the first process of central dogma. So, if you have not watched my video on central dogma, I recommend you to first watch that and then start with replication. I have made a very simple video on the concept with an example so that you can understand it easily. DNA is our genetic material. The most important characteristic of DNA is that it can make an exact copy of itself. And that is the very basis of life, and that process is called a DNA replication. Watson and Crick proposed the double helix model of DNA. During that time itself, they suggested the semi-conservative method of replication. So if we take their words, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. A scientist called Delbruck had of opinion that there could be three modes of replication of the DNA model proposed by Watson and Crick. Suppose this is the first parental DNA. First method is called a conservative method of replication, where one old parent strand will be like that. From that, a totally new strand will form. So, old strand is this green one and the red is the new one. So, old one is completely conserved and a new one is forming. Whereas, the semi-conservative method of replication proposed by Watson and Crick, the old strand separates out into two and in each one a new strand is forming. So, two strands of DNA would separate and act as a template for synthesis of new complementary strands, because when they separate out, there should be an order in which a new strand can form. That is called a complementarity. We already learned about base pairing. Adenine base pairs with thymine, guanine base pairs with cytosine. So, based on base pairing, a new strand is forming. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. Here, one of the parental strands is present in each new DNA. That's why it is called semi-conservative. Not completely conserved, but partially conserved. That's called a semi. Then there is a third method called a disruptive, where it's actually the parental DNA will fragment into many pieces and then just they replicate and randomly they join back. So, it may be like a random here and there, a red, like this. This is called a disruptive. There is no clear order. But we are going to learn about semi-conservative method of replication. Though Watson and Crick proposed it, this was confirmed by experiment done by Michelson and Stoll. Now let us see the experimental proof by Michelson and Stoll. It was done by Matthew Michelson and Franklin Stoll, or we call it as Michelson and Stoll experiment. They conducted it in the year 1958 and the experimental material was Escherichia coli. As you all are aware, Escherichia coli is a bacteria that multiplies every 20 minutes. So, one bacteria becomes 2 in 20 minutes, another 20 minutes, again that 2 will become 4, the next 20 minutes, the 4 will become 8, like that. So, first what they did, they grew the bacteria in a heavy isotope of nitrogen containing medium. That is, you know, normal nitrogen is N14. Then what is a heavy isotope of nitrogen? N15. So they grew the bacteria in N15 containing ammonium chloride. So we know when new DNA strands are being formed, they need nitrogen bases. So the nitrogen will be taken from the medium. So naturally what they are getting, only N15 is available, so they will be taking up only N15. So eventually in their body the DNA will form, the newly formed DNA will have N15. So these scientists, they separated out this heavy DNA and they used only the DNA containing N15 strands for their experiment. So here now it is a heavy DNA with isotope N15. Okay, so both the strands have only N15, no N14 or normal nitrogen is available. Now what did they do? They had to separate the N15 out of the mixture because there could be N14 one also. So they did centrifugation using cesium chloride. It was a density gradient centrifugation. Here we have to remember that N15 was a heavier isotope, not radioactive. So only when we do density gradient centrifugation, we get different layers based on their density. So heavier ones will be to the bottom, lighter ones to the top. So like that they separated it out. So when they are doing it the first time, they are getting N15 band at the bottom because all DNA strands have only N15. So it is towards the bottom, it's heavy. Then what happened? They allowed this DNA to grow in a medium containing normal nitrogen, ammonium chloride. Okay, normal nitrogen means what? N14 containing ammonium chloride. Now for the synthesis of new strand of DNA, what is the source available? Only N14. Whatever new strands they are making, the new strands have only N14 and the old strands have N15. So to see that, after 20 minutes, you know, every 20 minutes Escherichia coli is multiplying. So new DNA is forming. Okay, so what happens when they collected after 20 minutes, they again centrifuged it to separate out. So this time we know during DNA replication both the strands separate out and what happened? A new strand is forming. So a new strand is normal N14 which I am drawing using a green color. So here also the new strand is N14. So the old one is N15 and the new one is N14. So now when they are doing the centrifugation, what are they getting? A medium sized or in the medium band you will get. They get in the medium range. It's not heavy like N15 but it is a hybrid of N15 and N14. It's a hybrid, so it will be little above the heavier one. Okay, now again they waited for 20 minutes. So this time what happens? This will separate out. Both the DNA strands will separate out. Like this. So first one separates out. What do we get? One N15 in both the strands and one N14. But the medium has only N14, so the new one is invariably N14 or the green one. All are N14. Now if they do centrifugation, what will they get? The third case, that means totally after 40 minutes they will get, hybrid is still there, right? So a hybrid band is there with N14 and N15 plus what is forming? A lighter band, N14 only. Okay. Now again if you see up to 20 minutes now we did, that means total 40 minutes. Again after 20 minutes if you see what will you get? You know that the first one will be like this. There will be two red only, but here one green, 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 like this. Every single strand will make a new green one. So next generation, what are we expecting? We can expect one hybrid band plus majority lighter band. So again and again if you check after 20 minutes, it's going to be the same. More and more lighter bands will form, but still there will be a moderate or the hybrid band indicating that the old strand is retained. So with this experiment, Michelson and Stahl proved that DNA replication is semi-conservative. So during the same year, 1958, another scientist called Taylor and with his colleagues, that is Taylor et al., Taylor and his colleagues, they did experiment in faba beans. It's called a Vicia faba or faba beans. What is their mode of experiment? They used radioactive thymidine. We know that thymine is actually a nitrogenous base. So they used radioactive thymidine. So, and they could detect their distribution in the newly synthesized DNA easily being radioactive, so that they could confirm that the DNA replication is semi-conservative. What is replication? The unique process of making an identical copy of double-stranded DNA using existing DNA as template for the synthesis of new DNA strands. So here the newly formed DNA should be identical to the DNA that is existing. So we know that in our body trillions of cells are there. Every single cell has nucleus and it has DNA in it. So we all started from a single cell called zygote. So each time if the DNA is different, our cells would have got different types of DNA. It's not like that. Every single cell has an identical copy of our initial DNA. So it is very important that the parental strand or the old DNA acts as a template from which an exact copy is made so that the stability is maintained. Now, when we come to the site of occurrence of replication, in case of eukaryotes, we know that they have a well-defined nucleus and DNA or chromosomes are found within the nucleus. So, where is replication taking place? Of course, within the nucleus. But when we talk about prokaryotes, they don't have a nucleus. So, it happens in the cytoplasm. So, site of DNA replication is different for eukaryotes and prokaryotes. Now, coming to the phase in which it is occurring. We know DNA has to be doubled or the DNA has to replicate prior to cell division, because when one cell is becoming two daughter cells, equal amount of DNA should go to that. So without replication, if it is going, only half will be available to both. So suppose if this is a DNA and it has to go to two different cells, then it will become half and it will go to two cells. So each daughter cell is receiving only half the content of the DNA or half the information, so that won't exist. At the same time, if this is a parental DNA, it is making an exact copy of it prior to division. Then what happens? This is going to different daughter cells. So each daughter cell is getting a complete set of DNA. So it has to happen that the replication has to happen prior to cell division. You know, before actual division, there is a preparatory stage in the cell cycle called an interphase, where the cell is highly active. And the interphase has got again three substages: G1 phase, S phase and G2 phase. So in the S phase or the synthetic phase of the cell cycle, the DNA replication is taking place. This is very important. When does it occur? During the S phase. Now, where can it happen? Can it happen entire length of the DNA? We learned our DNA is very long. So, in case of prokaryotes, they usually have a single circular DNA. And in eukaryotes, it can be a linear DNA. So, in this case, the replication starts at a particular point. That point is called the origin of replication. So, if you look at the case of eukaryote, since the DNA is very long, it is not feasible to start from one end and continue till it reaches the other end. Whereas in prokaryotes, it's small DNA, so it can start from one end and it can continue, right, and complete. So in eukaryotes, there are several regions where origin of replication is there, or several regions where the replication can start, and it will continue and two will join. Just like if you have seen the repairing of roads or tarring of roads. If it is highway and all, kilometers of roads are there. So are they starting from one end and going ahead? No. You can see that work is going on at different places they start. And then finally everything will join. This is how it happens in eukaryotes. But prokaryotes, it is a single unit of replication starting from one end and ending in the other end. That means it's complete. So that unit of replication is referred to as replicon. So prokaryotes have a single replicon, whereas eukaryotes have several replicon. If you see DNA as a coiled structure like this, okay, helically coiled, and when this DNA has to unwind, because when it is in the form of a chromosome, suppose imagine this thread is a DNA which is highly condensed and formed in the form of a row-shaped structure called a chromosome. At this stage, the replication is not possible. So, first the double helical DNA has to unwind. So, how will it unwind? There are hydrogen bonds. By breaking the hydrogen bonds, it can unwind. So, once it is unwinding, this part is forming, or lower part, both the sides, there is a Y-shaped structure forming, that is called a replication fork. So actually replication is starting within that replication fork. So where is the replication fork forming? In the origin of replication. So what are the requirements for DNA replication? First and foremost, a DNA template. Because from nowhere we cannot make a new DNA. It should be based on the information on an already existing parental DNA. So template DNA is the first requirement. And then RNA primer is required. Primer means it is a small stretch of RNA. So it's not DNA actually, it is RNA. A small stretch of RNA is needed for starting the process, because the whole process is catalyzed by an enzyme called DNA dependent DNA polymerase. So DNA polymerase means what? The enzyme that can polymerize a particular reaction. Here polymerization is taking place. Nucleotides are added to make a new polynucleotide. So it is polymerization reaction. What is being polymerized? DNA. So it is called a DNA polymerase. But where is the information for making that polymerization? It is on a DNA strand. So DNA dependent DNA polymerase. Based on the information on a particular DNA strand, a new polymerization is happening. That's why the name is like this, DNA dependent DNA polymerase. So though this enzyme is the one making a new strand, this enzyme is incapable of initiating the process. It can just continue only. So there should be an RNA primer to start the process. Okay. Then next is the raw material. You want to make a new strand. How can we make a new strand of DNA? Using nucleotides, right? So we need nucleotide as the substrates. They are present in the cell. There is nucleotide pool there. From there DATP, DTTP, DGTP and DCTP will be taken according to complementarity rule and will be attached. That is, if on the template strand, if there is A, T, G, C. So the DNA replication, new strand is forming. Here T will keep, here A, here C, here G. So it can be made. But these nucleotides have to attach with a phosphodiester linkage. For that energy is required. Who will give the energy? The same substrates will give energy. So they have dual roles. One, they act as the substrate for the replication process. Second, they provide energy for polymerization. Okay, this is a very important question. What are the dual roles played by the deoxyribonucleoside triphosphates? They are energy rich compounds. Then coming to the various enzymes required. A lot of enzymes are there helping DNA dependent DNA polymerase. So first is DNA unwinding protein or helicase. Suppose this is a DNA strand. It has to unwind first. So we know that these two strands are connected by hydrogen bonds. So first hydrogen bonds have to be broken. So who will break that? There is an enzyme called a helicase, or we call it as unwindase enzyme. Okay. So first DNA helicase or unwindase enzyme separates the two DNA strands, not at the entire length, at origin of replication. So what are we getting? A Y-shaped fork. On both the sides of origin of replication, we are getting Y-shaped fork. So now we know that once this part is unwinding, there is a tension created here, or this part is undergoing supercoiling, both the sides, right? Supercoiling. So that tension has to be released. For that, there is another enzyme in eukaryotes called a topoisomerase. So what does it do? it has the ability to make a nick. Nick means what? Cut. It will cut this strand, okay, then unwind it and then join using another enzyme called a ligase, so that tension is released. One cut, unwind, join back, so tension is released. So that supercoiling tension is relaxed by the topoisomerase in case of eukaryotes. When it comes to prokaryotes, for them the opening of the helix and the tension release both are done by a single enzyme called a gyrase. Okay, so hope that is clear for you. Now, the third enzyme is RNA polymerase. As I mentioned, DNA dependent DNA polymerase can only make the new DNA strand, but for starting RNA primer is required. So who will make RNA primer? RNA polymerase enzyme. Then the last is polynucleotide ligase. Finally, after the process is over, RNA primer has to be removed, and in that place nucleotides will be added based on complementarity. Then that has to be attached using DNA ligase enzyme, and one more function is there for DNA ligase, joining function only, that we will discuss when we are talking about the replication process. After this enzymes, there are proteins required for this process, protein factors. That is, if this strand is separating out, but these strands are complementary, so there is a chance that they can come and join back. So to prevent that, there are certain proteins coming and attaching here on the sides, both the sides, called a single strand binding protein, because they bind to a single strand so that it will prevent it from rejoining. So protein factors are also required for this process. So this is the replication fork. We know that there are two strands which are separated now. This is 3' to 5'. The other one is 5' to 3'. So we know that DNA dependent DNA polymerase enzyme is the enzyme that catalyzes this whole process. So, but the new strand is formed only in 5' to 3' direction. This is very important. You remember it as a thumb rule. Always a new strand forms in the 5' to 3' direction, or the polarity of the new strand is 5' to 3'. Okay, whether it is replication or transcription, the same rule follows. Then here, you know, if a new strand is 5', we know that both the strands of DNA are anti-parallel in polarity, right? So this is the 3' end. So here only 5' can come. Here a 3' cannot come, right? Because it is going to be a new DNA. So this here it should not come. First, what will happen? An RNA primer should come and attach. So where will it come and attach? It is in the 3' end. 3' end, a short stretch of RNA is coming, which is having 5' to 3' polarity, right? Because anti-parallel. For this strand, it is like this. Now, in this strand also, both the strands act as template. Okay. So here, can the RNA primer attach here? No, because the new strand is only in the direction 5' to 3'. So here to 5' to 3' is not possible. This both will be 5'. So where is it available now? Here. So here is a 3' end. Up to this is closed. So only open part, the new one can form. So for the time being, it's starting from here. Okay. So this is the primer. So it will have 5', 3'. Now there are nucleotides. Suppose I am just writing this is, and single strand binding proteins will be attached here to prevent the joining back. So suppose this is T, A, G, C, C, T, T, A, G. I'm just randomly telling. So we know that this side it will be, this is complementary, right? So this will be C, T, T, A, C, sorry, G, G, C, T, A. So complementary. Now what will happen? The new one, first what will come? It is T. So what should come? A. It will be similar to this strand because it is complementary. So here A will come, then T, C, G, G, A, T, C. So this way it will proceed with the help of DNA dependent DNA polymerase. The DNA polymerase enzyme will bring DATP, DGTP, DCTP, whatever is required here, and attach, and with the substrate energy they will form the bond. The same way here also complementary strand is forming G, A, A, T, T, C, C, G, A, T, 3 prime. Okay, so whatever is open, that part is done. Now, this strand reached here up to this open end. Now, if it has to proceed further, more opening should form, right? More unwinding is required. Meanwhile, this part will form the helix. So now, if we unwind it further, I will just show you like this. Again, unwinding. Okay. So this part almost reached here, maybe 3 prime, 5 prime, 3 prime. This part also 5 prime to 3 prime. Okay. Now more unwinding. So that I will enlarge and draw again. So this is how it is. This is RNA primer, here the RNA primer. Now it has to proceed. So proceeding in the direction 5 prime to 3 prime. So it's already 5 prime to 3 prime. So it can go beyond, right? Now this part is open. Before this part was not open. Now this part is open. So it will continue this way till here. Based on complementarity. So there is no need of RNA primer. But in this strand there comes a problem now. Why? Because it cannot go back. The new strand is from 5 prime to 3 prime direction only. So it started from 5 prime, it went to 3 prime. Now backwards it cannot go. So again a new synthesis should start. So another RNA primer will come to that 5 prime end. Okay. From there a new one will form. So now if it is unwinding further, more has to unwind. So this part again 5 prime it will go continue, but here already 2 stretches have formed with RNA primer. Then now again it has to start from here. Okay, so in this strand it is continuous. Where is it continuous? The template strand with the polarity 3 prime to 5 prime, the replication is continuous. Whereas on the template strand with the polarity 5 prime to 3 prime, it is discontinuous. So this strand is otherwise called the leading strand. Because it's a single stretch going on. Now here there are many fragments that are forming. These fragments are called Okazaki fragments. Okazaki fragments, and the strand is called a lagging strand. So here synthesis is continuous, discontinuous synthesis. Strand is called a leading strand, here it is called a lagging strand. Lagging strand has many fragments of DNA, they are called Okazaki fragments. But now we want an entire DNA, right? This is the diagram of a replication 4 given in your textbook. It is important diagram, though it looks simple. Whenever you draw, make sure that you are writing the polarity correct. Okay, so the new one will be, leading strand will be 5 prime to 3 prime. So here now what happens? Here we know a complete DNA strand we will get, but here we are getting fragments. But we want the DNA to be complete. Now the next stage is removal of RNA primer. Actually, DNA polymerase 1 enzyme will come and degrade this RNA and form the DNA strand based on the complementarity on the parent strand. Here also, wherever gaps are there, those gaps will be filled. So as a result, there will be a continuous strand this side also, and for joining these newly formed fragments into one, they use another enzyme called a polynucleotide ligase enzyme. So always ligase enzymes are the molecular glue sticking together the parts of nucleic acids. So here ligase is used to join. Now both the strands are complete. So from one DNA double helix structure, now we got two DNA double helices. So this is the process of DNA replication. Now another amazing property of DNA is DNA repair. When the new strand is forming based on complementarity, there can be errors. So the DNA polymerase enzyme will be going through the strand to check for any damage caused or error occurred. If there is an error, that will lead to mutation. DNA polymerase tries to reduce the errors to the maximum level. So during proofreading, suppose there is a wrong or a mismatched DNA strand, or there is a nitrogen base which is wrong. So using nuclease enzyme, it will make the nick or cut it, remove it and replace it with the correct one and join using DNA ligase. So this kind of proofreading is done throughout the DNA during replication so that errors are minimized. And as and when these strands are forming, they again coil to form the double helix DNA. Apart from this, then the damage repair system is also present in our cell. If the cell is undergoing replication, but after that the cell is not dividing. Now the nucleus has double the amount of DNA, but chromosome number remains the same. That is, if it was like this, if the chromosome was like this, after replication what happens? It has one more arm. But it is not a new chromosome, the same chromosome DNA content doubled. Okay, number is not increasing. So this will lead to a condition called a polyploidy. A failure of cell division after DNA replication will lead to a condition of polyploidy where the amount of DNA will be more than the actual amount. In 1956, a scientist called Korenberg, he discovered DNA polymerase enzyme. Then in 1959, he made a new DNA strand in vitro, means in the lab, or artificial DNA strand was formed using dNTPs and DNA polymerase enzyme. Hope you understood DNA replication process. Please like, share and subscribe to my channel, Biology My Passion.